12 will be about protein sorting. So first we'll have an overview of protein sorting. 2, various peptide sequences that enable sorting. 3, events in the endoplasmic reticulum and Golgi. We'll talk about the signal sequence, targeting to the endoplasmic reticulum, end-link glycosylation in the ER and Golgi, transport in secretory vesicles, mannose phosphate, and lysosomal proteins. In part 4, transport to mitochondria. Part 5, transport to the nucleus. And part 6, protein degradation. Ribosomes participate in protein synthesis as free or membrane-bound complexes. This determines where the proteins go and how they are modified and what they do. This is an overview of protein sorting. It's a pretty good picture. So here's our messenger RNA. The messenger RNA can go in 2 directions. It can be translated as a free messenger in the cytosol, or it can end up on the rough endoplasmic reticulum and synthesize proteins that are destined for the luminal part of the endoplasmic reticulum. So let's start with mRNA going to the rough endoplasmic reticulum. So we're going to have a nascent protein. This is the protein that's being synthesized, and it's going to have a polypeptide sequence on it that's going to be the endoplasmic reticulum signal sequence. And then there will be a protein complex that binds to this sequence, and it will attach the ribosomes and the messenger RNA to the endoplasmic reticulum. We'll process these proteins in the endoplasmic reticulum, and then they will transverse the Golgi complex or the Golgi apparatus, and then after all the modifications and the things that happen in either the endoplasmic reticulum or the Golgi complex, we will have cell surface secreted proteins. We'll have proteins that go to the lysosomes and proteins that are inserted into the plasma membranes. So that's the rough endoplasmic reticulum story. Freestanding mRNAs that are being translated in the cytosol. We can make cytosolic proteins. For instance, most of the enzymes of glycolysis are cytosolic proteins. So we synthesize the polypeptide chain folds, and then the protein is functional in the cytosol. Or we can have an organelle-specific uptake targeting sequence. This yellow box represents a targeting sequence, and from there, the protein can be imported into the mitochondria. It can be imported into chloroplast. It can be imported into peroxisomes, or it can be imported into the nucleus. So these are all done from freestanding mRNAs and protein synthesis, whereas over here we will have a signal sequence that will target this particular mRNA that's being translated onto the surface of the endoplasmic reticulum. Some typical signal sequences. So the function of the sequence, import into the nucleus. And the signal sequence is proline, proline, lysine, 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 arginine, lysine, valine. So lots of basic amino acids in here. Plus you start with proline. Export from the nucleus. Leucine, alanine, leucine, lysine, leucine, alanine, glycine, leucine, aspartate isoleucine, many hydrophobics, leucine, alanine, leucine. Here's a basic leucine, leucine. Import into mitochondria. This is the amino terminal N. Here's a methionine, leucine, serine. Don't need to read all these. That is a signal for import into mitochondria. Import into peroxisomes. On the carboxyl terminus, we have serine, lysine, and leucine, just 3 amino acids, and that targets these particular proteins into peroxisomes. Import in the endoplasmic reticulum, we have our signal peptide, and it's variable but very hydrophobic. So here's our methionine, which was our first amino acid that was incorporated into the growing chain. Just like this is the methionine, that's the first amino acid that is incorporated into the chain. Here's the amino N, methionine, lysine, alanine. So this is a basic 4 basic amino acids or so, and then we have a long string of hydrophobic amino acids. Notice leucine, 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 valine, glycine, isoleucine, leucine, valine, phenylalanine, glycine, asparagine. So these are either hydrophobic or nonpolar. And then the return to the endoplasmic reticulum, this is where the protein is now not in the endoplasmic reticulum, but the particular sequence lysine, aspartate, glutamate, leucine on the carboxy terminus will target particular proteins for import into the endoplasmic reticulum. So they've already left the endoplasmic reticulum and now they're coming back. This is the sequence that puts them back into the endoplasmic reticulum. Membrane-bound ribosomes. So let's start with our messenger RNA. Here's our 5' cap. We've done all of our splicing. How do we know? Because we're in the cytoplasm and we had to do our splicing correctly to go through the nuclear pores. Here's our start signal, UAG. The signal peptide is towards the N-terminus. It's only about 2 or 3 amino acids removed from the N-terminus, so we generate our signal sequence very quickly. We have a signal recognition protein, and it recognizes the signal sequence. And now the ribosome and the signal recognition peptide and the signal sequence binds to a ribosome receptor. It's called the signal recognition protein receptor, and all of these bind in this complex. And now we continue our protein synthesis, and notice polypeptide chain is getting longer, and it's basically going through a pore. So you could think of it as being injected into the endoplasmic reticulum as the chain is being synthesized. And now we have what's called the signal peptidase, and it will cleave off this signal part. And then what happens to the protein at this point depends on its sequence. In this case, they're showing a protein that's folded and it's in the lumen of the endoplasmic reticulum. So we have a receptor that's going to recognize the ribosome, and it's going to recognize the signal peptide, and it's going to recognize the signal receptor protein. We get a complex, and the protein synthesis basically injects the protein into the ER lumen. So these are signal peptides from different organisms. So from influenza, here's our hydrophobic stretch. We start with methionine. Remember, that's our terminal amino acid. Lysine, alanine, lysine, 2 basics. And then we have our hydrophobic stretch. This site that's shown with the arrow, that's where the signal peptidase cleaves. So once we cleave it off, the amino terminus of this particular protein will be an aspartate, not a methionine. Here's human pre-pro insulin. So here is our basic stretch. There's our basic amino acid. Here's our hydrophobic stretch. This is where we cleave off the signal peptide, cleave it off here, methionine, etc. So these all show the sequences aren't exactly the same, but what is the same is 1 or more basic amino acids before the signal sequence, and then the signal sequence itself is a hydrophobic stretch of about 8 to 10 amino acids. This is rough endoplasmic reticulum, the part right here between this membrane and this membrane. This is the lumen. This is a high number of ribosomes on the endoplasmic reticulum to make the rough endoplasmic reticulum. This is rough endoplasmic reticulum from pancreatic cells. And remember, pancreatic cells make high levels of either peptides or proteins. Alpha cells make glucagon that they secrete. Beta cells make insulin, which they secrete. The exocrine cells make hydrolytic enzymes, chymotrypsinogen, trypsinogen, proelastase, procarboxykinase, the different lipases. I would assume with the number of ribosomes that it's a good bet that these are pancreatic exocrine cells, and they're synthesizing hydrolytic enzymes for secretion. Formation of N-linked glycoproteins in the endoplasmic reticulum and the Golgi. So many proteins that are synthesized in the endoplasmic reticulum via this process of attaching the ribosome and making rough endoplasmic reticulum. Many of those proteins are glycoproteins. So let's start with the rough endoplasmic reticulum. Down here is our mRNA, 5' N, 3' N. Here's our growing protein that's being distributed into the lumen of the endoplasmic reticulum. By N-link, carbohydrates are linked to asparagine. So now here's our rough endoplasmic reticulum. Here's our protein. That's a glycoprotein. Now let's go back to step one. We're going to make a glycolipid tree. And dolichol is a lipid, and it's made out of isoprene units, and it has a phosphate group on it. Here's the phosphate group, and this part in here is hydrophobic. And then we take two UDP N-acetylglucosamines. GLC-NAC means N-acetylglucosamine. Blue square, and this blue square represent N-acetylglucosamine. And we have a pyrophosphate linkage here. One of the phosphates came from this phosphate. The other phosphate came from the UDP. And then we take 5 mannoses from 5 GDP mannose. So this is the activated carrier of mannose, GDP mannose, just like UDP N-acetylglucosamine is the activated carrier of N-acetylglucosamine. So we take 5 of these. Here's our 5. They're in green. Remember, our N-acetylglucosamine is in blue. And then we translocate this entire process and we flip it around where the hydrophobic part's on the outside and this glycolipid tree part that we're synthesizing is on the inside. And this is extraordinary. This translocation is extraordinary. We have a large protein complex that actually flips this molecule from inside orientation to outside orientation. So right here, we've added 4 mannoses. Here we've added 3 glucoses, and then we transfer this entire molecule, this part right there, we transfer it to asparagine. So that's called our N-link asparagine. That's transferred to the amide nitrogen of asparagine, and the PP group stays on the dolichol. Here it is, it's on the dolichol. So we transferred it. This is called an N-block transfer. And by N-block, it means we make the glycolipid tree first, and then we transfer the whole tree to the asparagine. Here is our glycoprotein that we synthesized. And now we can recycle our dolichol pyrophosphate. So we convert dolichol pyrophosphate and dolichol phosphate, and then we start this process again. We go over here, we flip this over where the carbohydrate part is now on the lumen side rather than the cytosolic side, and then this glycoprotein that we make can go to the Golgi. So there's many things that'll happen after we've made this initial glycoprotein, and the part that's shaded in sort of the yellow, that's called the core carbohydrate, and many of these other carbohydrates will be removed. Pathway taken by proteins destined for lysosomes, the plasma membrane, or secretion. So here's our rough endoplasmic reticulum. You can see the ribosomes, and then we make transport vesicles. So we pinch off transport vesicles from the endoplasmic reticulum, and they fuse with the Golgi apparatus. So this protein that we made, it's going to the Golgi apparatus in a vesicle. So these structures are going to enclose these glycoproteins in a membrane sphere, and then it's going to be transported to the Golgi apparatus, so the Golgi stacks, and we'll have additional modifications happening here. And then we will pinch off more vesicles, except we're going to call these vesicles secretion vesicles. So these are transport vesicles. This is called the cis side. This is called the trans side of the Golgi. And this picture shows the same thing. Here's the nucleus. Here's the rough endoplasmic reticulum. Here's our transport vesicles that have protein in them. They fuse with the cis side of the Golgi, with the Golgi complex. And then we start our sorting on the trans side. Some of these proteins will go to lysosomes. Some will go into secretory vesicles and get secreted. This is what happens with our exocrine pancreas and the secretion of the hydrolytic enzymes. The spherical structures fuse with the membrane and release their contents. And then some of the transport vesicles that have proteins that are inserted into the membranes, they will fuse with the plasma membrane. Mannose 6-phosphate pathway targeting lysosomal enzymes to the lysosomes. So here's our rough endoplasmic reticulum. We've made glycosylated proteins, so these are glycoproteins. Some of these proteins will become secretory proteins. Others will become lysosomal enzymes. Others will insert into the membrane and become membrane proteins. We phosphorylate mannose, and we're going to do this in a very unusual way. So remember, we had a whole bunch of mannoses on these glycoprotein trees. Let's go back and look real quick. All of these things in green are mannoses. So we have many mannoses that we can phosphorylate. So we're going to phosphorylate a mannose, and then the phosphorylated mannose targets this particular protein to the lysosome. If the proteins aren't phosphorylated, they're going to go into transport vesicles, and they will either be secreted or fused into the membrane. So if you phosphorylate a glycoprotein, it's going to the lysosome. If it's not phosphorylated on mannose, it's either going to be a secretory protein or it's going to be a membrane-bound protein. So here we're dividing the rough endoplasmic reticulum. Actually, we're going through the Golgi now. We're dividing it into two parts. We have a mannose 6-phosphate receptor that's going to bind to the mannose phosphate, and then we're going to pinch off this protein in a vesicle, and really there'll be different mannose phosphorylated proteins in this vesicle, not just one, but this shows it as one. We lose the clathrin coat. The clathrin coat enables the formation of these vesicles. It basically puts a protein coat around the vesicle. And now we lose our clathrin triskeletons. Here's the uncoated transport vesicle. It fuses with endosomes. Endosomes are structures that are being endocytosed, and when the uncoated transport vesicle, the endosome fuses with the uncoated transport vesicle, and now we have proteins and other structures that are going to be degraded, plus our hydrolytic enzymes, the proteases, the lipases, and other type enzymes. The galactosidases, for instance, that we saw in Tay-Sachs disease, will be in this endosome. We fuse this with the lysosome, and now we have our acidic compartment, and also the proteins that were in here from endocytosis. They may be in the lysosome, and if they are, they will be degraded. So remember, we started with a mannose 6-phosphate receptor. The mannose 6-phosphate receptor gets recycled, and it can go back to this particular Golgi type structure, or it can go to the plasma membrane where it can capture other mannose phosphorylated enzymes, and they can come back into endosome. And also we will have receptors for other kind of proteins that are being recycled and end up in these endosome structures, which eventually, after our sorting, some of them will end up in our lysosome. So very, very interesting complex structure. If you don't phosphorylate the mannose, these hydrolytic enzymes don't end up in lysosomes, and you can have all sorts of pathological problems. Phosphomannose targets hydrolases for vesicles that fuse with lysosomes. So that's what we saw back here. Here's our vesicles, and eventually these hydrolases end up in the lysosomes. But we're not going to phosphorylate mannose by a kinase. This is kind of a twist. So here's our enzyme that has the oligosaccharide, that has the mannose residue, and it reacts with UDP in acetylglucosamine. And what it will do is it will add a carbohydrate through a phosphate group. So usually we make glycosidic bonds that are between carbons and oxygens and carbons, but in this case we have a phosphate linkage. Then we have a phosphodiesterase that will remove the N-acetylglucosamine and leaves the phosphate group on mannose. So we phosphorylated mannose not by kinase, but by making a phosphodiester linkage between N-acetylglucosamine and mannose, and then we remove the N-acetylglucosamine, and that leaves a phosphomannose. Really interesting phosphorylation mechanism. And here's our enzyme that's going to the lysosome because it has phosphomannose on it. Import of proteins into the mitochondria. Here is the precursor protein that's going to have a signal for matrix targeting sequence. And the matrix is the mitochondrial matrix. That's the area in here. So a nascent precursor protein with the matrix sequence, it's recognized by the mitochondrial import stimulating factor. MSS is mitochondrial import stimulating factor. And so obviously we'll make a complex. We take ATP and hydrolyze ADP and inorganic phosphate. And now this binds to a complex of proteins that are called TOM. And we have TOM 70 and Tom37, and this is on the outer mitochondrial membrane. Remember, mitochondria are a double membrane system. We have the outer mitochondrial membrane and the inner mitochondrial membrane. And Tom is the transporter of the outer membrane. So Tom is on the outer membrane. This MSF and the protein bind. MSF leaves. The protein's bound to the transporter. There's a Tom40 that's a pore, and it gets transferred to the pore, and it goes into the inner membrane space. So that's method 1, the mitochondrial import stimulating factor. Method 2 is a cytosolic heat shock protein 70 that binds to the polypeptide, and notice it unfolds it. So if it's folded here, now it's linear. It delivers, with the hydrolysis of 2 ATPs, it delivers the peptide to Tom70, Tom37, Tom40 comes in, and now it's imported into the inner membrane space. Tom40 is actually in contact, it doesn't show this, but it's in contact with TIM44 and TIM2317. And TIM is the transporter of the inner mitochondrial membrane. So we have a transporter that brings it through the outer membrane and a transporter that brings it through the inner membrane, and now we're in the matrix. So Tom outer, TIM inner. It's believed that what drives this through here is the proton motive force. Remember, we will have a proton gradient, and it's believed that that gives us the energy to drive this protein through these pores. And then heat shock protein 70 binds again. Remember, it bound up here, and it basically straightens out the polypeptide chain. It binds here, and it keeps the protein from folding as it enters the mitochondria. So you want it all to enter the mitochondria before it folds. So here it is. Now it does its folding, and we have the active protein. We can also use heat shock 70 again, an ATP, and we can get an active protein. So these heat shock proteins are involved in polypeptides folding correctly. So it's in here. Maybe it folds correctly without it. If it doesn't, the heat shock protein will help it fold correctly. Most of the proteins that are in the mitochondria weren't made by the molecular biology of the mitochondria. Its DNA, its RNA, its protein synthesis only makes about 10 or so proteins, and all the rest of the proteins are in the mitochondria through this particular mechanism, and they're targeted to the mitochondria through this matrix targeting sequence. So this is a lot like the signal peptide that targets a protein for the endoplasmic reticulum. This sequence targets the protein for the mitochondria. Targeting of nuclear proteins. None of the proteins in the nucleus were synthesized in the nucleus. They were synthesized on free ribosomes and mRNA in the cytoplasm. This will be a nuclear protein. NLS means nuclear localization sequence. NLS, nuclear localization sequence, and this is the sequence on the protein that's going to target it for entry into the nucleus. And we have particular proteins called importans. So we have an alpha and beta important that's a dimer, and it binds to this sequence. The important protein complex can be imported through the nuclear pore, so this represents the nuclear pore. And when we enter the nucleus, the importants are going to dissociate. B dissociates to a protein called RAN that has GTP on it, and RAN means RAS-related nuclear protein. And we'll find out a little bit more about that later. So RAN, RAS-related nuclear protein with the beta subunit. And now we have the protein with the alpha. The alpha dissociates, and here's our protein that will become whatever it's supposed to be. Maybe it's a histone, transcription factors, one of the DNA polymerases, replication factors, many things. Thousands and thousands of proteins are represented by this polypeptide chain. And then when the alpha dissociates, binds to a protein called Cas and RAN again. So RAN binds both the beta and it binds both the alpha. And Cas stands for cellular apoptosis susceptibility protein. The RAN GTP with beta, the RAN GTP with alpha and Cas, they all get transported out of the nucleus. And now here's our alpha on the outside. Here's our beta. The beta and the alpha can associate with another polypeptide chain and be imported back into the nucleoplasm. The Cas dissociates, So this represents RAN coming up here. We had two of them. We had a RAN here, RAN GTP, RAN GTP. The transport through this particular pore involves hydrolysis of GTP to GDP, an inorganic phosphate. So here's our alpha, here's our beta. They're back over here. We mentioned that. And the RAN now has a GDP. In order to use it again, we need to convert it back to the GTP. So we have NTF2, which is the nuclear transport factor 2, and it binds to the RAN GDP. It brings it through the nuclear pore, and now we convert the RANs through a GDP GTP exchange factor. Here's the GTP, here's the GDP. Now this has GTP on it. We can convert the RAN into its active form that can now be used for transporting alpha and beta importance back into the cytosol. This is a complex picture. Polyubiquitination specifically targets protein for destruction by the 26S proteasome. So we have several mechanisms that have to do with the rate at which we destroy protein molecules. So when we make protein molecules, they don't last forever. They have a particular life. It's called a half-life. So ubiquitin is a small protein, and it's called ubiquitin because when people started to analyze this, it was found in almost all cells. So it was considered to be ubiquitous. So it's called ubiquitin, and here's the carboxyl N. E1 binds ubiquitin through a process that takes ATP, and it binds through a sulfhydryl. So here's E1 with its sulfhydryl, and here's ubiquitin bound to it. And now E1 transfers this ubiquitin to E2. This doesn't require energy. So here's an exchange basically between E1 and E2. So now here's E2 that has the ubiquitin on it, and then this ubiquitin is transferred to the target protein. And many of these target proteins, they're target proteins because they're defective. So defective proteins are targeted by ubiquitin, and that is the signal for degradation because we don't want defective proteins, we want functional proteins. And the enzyme that does this transfer is called E3. So we have an E1 that accepts ubiquitin. E1 exchanges its ubiquitin with E2. E2 transfers the ubiquitin to the target protein in a reaction catalyzed by E3. So it's polyubiquitination that causes the degradation. So we don't add one ubiquitin, we add many ubiquitins. And then the degradation is going to happen in a structure that's called the proteasome. So the proteasome is a hydrolytic structure. It has hydrolytic activity in this barrel. It looks like a barrel if you see it in 3 dimensions. And the proteasome destroys ubiquitinated proteins. So here's our substrate protein. Here's a polyubiquitin that's recognized, and this is basically transported into this barrel, and the barrel hydrolyzes the structure dashes. Those are individual amino acids. And then we also have a regulatory protein. So polyubiquitin attached to protein interacts with the proteasome. This shows the degradation, and the regulatory protein is going to control how active we are in degrading proteins. Relationship between protein half-life and the amino-terminal amino acid residue. So ubiquitin is one mechanism for destroying proteins that are structurally defective. And what makes them structurally defective? How do you determine that? If they're not folded right, they will have groups that are exposed that aren't usually exposed, and they become targets for the ubiquitination. So stabilizing amino acids on the end. Here's our MET. This was the first amino acid. As we saw previously, not all proteins are going to have MET at the end. For instance, if you have a signal peptide, the MET part gets removed when you remove the signal peptide. A glycine, an alanine, a serine, a threonine, a valine, all these stabilize the amino end of a protein, and you have half-lives of more than 20 hours. So a protein, half of the proteins will last for 20 hours or more. That's what we mean by half-life. Destabilizing. Isoleucine and glutamine, approximately 30 minutes. So if the terminus is not one of these, but isoleucine or glutamine, half of the proteins will be destroyed within 30 minutes of being synthesized. Tyrosine, glutamate, 10 minutes. Proline, 7 minutes. leucine, phenylalanine, aspartate, lysine. 3 minutes. Half of them after synthesis will be destroyed within 3 minutes. Arginine within 2 minutes. So this is just fascinating. The amino acid on the end of a protein that's fully functional and is not misfolded. It's not defective. Terminal amino acid is arginine. Destroyed. Half will be destroyed within 2 minutes. So relationship between protein half-life and the terminal amino acid residue. So this ends L12, and it's also the last presentation in Chemistry 256.